All right. Good evening, guys. I hope you're all doing well. Hello.
Hello.
Hi, madam.
Hello, madam. We are here.
Yeah,
This
around
is
now.
Hi, madam. We are here.
yeah.
Hello, madam.
Hello, hello. We are here.
Hello, madam. We are here.
Hello, madam. Good evening.
Hello.
All right. You're welcome once again. This physiology. Where did we get to? I'm sure. I hope you can see the slide. Is that where we got to the other day? Okay. You have forgotten. I think this is where we got to the other day. The fourth level of organization, which is the system. I believe the class can hear me.
Madam, please, we can hear you.
Yes, please, sister, we can
Yeah,
hear
we
you.
can hear you, please, madam.
Yes, please, we can hear you.
All right. Then let's start. Edward, when you mute the class, you mute me also. So just leave it to me. Let me control the class, okay? Okay. So the system level. A system is made of two or more organs that are performing a similar function. So you can have a lot of organs within the human body, but some will come together to form a system based on their function. Examples of such systems, you have digestive system, respiratory, cardiovascular, urinary, integumentary, name them. They are 11 plus the special senses. So you have to, for now they are 11. You are going to take each one of them one after the other and then look at how they function from the cellular level. Then finally, we have the organism as the fifth level of organization. An organism is a collection of all systems together working to ensure homeostasis. You have 6 important life processes: metabolism, breakdown of substances and formation of molecules within the body; responsiveness, your ability to respond to change, stimuli, main function of the nervous system. You can also talk of movement, can talk of growth, differentiation of cells, and maturity of cells. You can talk of reproduction, continuity of life. So in health, all parts of the body must be functioning together in a process called homeostasis. If you are sleepy, kindly wake up. This is the last class for the day. Let's enjoy it. I know you are tired. Just allow yourself some few hours. You'll be through. All right. So as a human being, we have survival needs, and these survival needs are essential for your systems to function well. First of all, nutrients. Nutrients is gotten from the food we take in: carbohydrates, fast proteins, minerals, and vitamins. And these components are broken down within the body. They are simpler units absorbed, and the cells will use these simpler units to further produce body proteins and so on. Then we also have oxygen, essential for ATP production. Water is also life, which helps with chemical reaction. And then we also have another survival need, which is normal body temperature, essential for cells and tissue optimal function. Then we also have an appropriate atmospheric pressure that is essential for gas exchange in the lungs. Model 2 of Unit 1, we want to look at body fluid compartment. But the body fluid compartment basically tells us the various chambers within the body that have fluids. And when you talk of fluids are just water plus dissolved substances. These dissolved substances can be ions, they can be proteins, they can be fats, they can be different kinds of molecules that are found or that are dissolved in water that is found in the various chambers within the body. We want to look at them. We have two main components of body fluids compartments, or two main types. You can talk of the intracellular fluid and the extracellular fluid. Intracellular fluid is the fluid found within the cell. Within. Intra is within. Cellular is a cell. So within the cell, the fluid found there, and that's the cytosol. Then we also have the extracellular fluid, which is the fluid that's found outside the cell. Under the extracellular fluid, you can have some subtypes. You we can talk of the fluids that surrounds right around the cells. So just around the cells, we have fluids, that is the interstitial fluid. Then we have plasma, which is the fluid part of the blood. That is the main means of transport within the body. Then we have transcellular fluids. Transcellular fluids, these are fluids aside the plasma and then the interstitial fluids. These are the extracellular fluids aside the plasma and then the interstitial fluids. Example is the lymph found within the lymphatic vessels. You have the cerebrospinal fluids found within the brain and then the spinal cord. You have the synovial fluid found in joints, in between bones, joints. And then we have aqueous humor found within the eye. All these fluids form transcellular fluid compartment. So to briefly look at how transport of substances and molecules would occur across the different kinds of fluids or the fluid compartment, we can look at the lung, which is the organ that aids in breathing. So when you breathe in, the components of air, for example, oxygen, diffuses into the blood, which is an extracellular fluid. Then the blood will transport the oxygen to the cells and tissues, but first of all, it diffuses into the fluid surrounding the cells, which is the interstitial fluids. Then eventually, this oxygen also diffuses into the intracellular fluid. Metabolism occurs, carbon dioxide is produced as a byproduct. Carbon dioxide diffuses out of the cell into the interstitial fluid, eventually diffuses into the blood. Then blood takes it to the lung level where you expire the air. At the GI tract, we also have nutrients absorbed within the small intestines, then into the blood. Blood transports these nutrients to the cells, but first of all, they are diffused into the interstitial fluid and eventually into the cells. Same for water and so on, ions. Then we also have nitrogenous waste that is produced by the cells at the end of metabolism. Then they will leave the cells into the interstitial fluid, into the blood. Then the blood takes it into circulation, where when it gets to the kidney, it's filtered to produce urine. So this is among the few how some components within the body are moved in the various fluid compartments. Any questions up to this point? Any questions up to this point? No question? Well understood. All right. Then we look at total body water. Total body water is very essential and it needs to be maintained by homeostasis. We talk more about volume of water, homeostasis of volume of water when we get to the urinary system. We want to look at the different percentages of total body water in individuals. We have some factors that influences differences in total body water. First one is age, second one gender, third one is the degree of obesity. Age. Why is it that you have different volume of water in an adult compared to an infant or a child? Why is it that males tend to have more body water compared to females? Why is it that someone who is obese has less volume of water compared to someone who is lean? This is a reading assignment that I want you to take note. Accounts for the differences among the three with respect to total body water. Then we look at Unit 2. So we are done with Introduction to Physiology, the different level of organization, and then the body fluid compartment. Unit 2, we want to look at the cell. the plasma membrane, and then the various transport mechanisms. Model 1 will focus on the cell or plasma membrane, the structure of the plasma membrane, and how it's going to facilitate the different transport mechanisms. The plasma membrane is flexible, it's not rigid, it's very flexible, and that is as a result of the components. First of all, you can look at the abundant component of the cell membrane, which is the phospholipids. Phospholipids are molecules that have a polar end and then a non-polar tail from the slide. So we have one molecule of a phospholipid, has a polar head, non-polar tail, and is arranged in two layers, the top one, the bottom one. So the top one faces the external of the cell, the bottom one faces the inside of the cell. And it creates a barrier between the extracellular and then intracellular fluid. Barrier meaning it regulates what enters and leaves, just as we have a checkpoint or a barrier when you are traveling. So it checks what comes in and goes out. It limits movement of some particles in and out, and it also allows others to pass through. The cell membrane is made of phospholipids. I've mentioned that, and it has two polarity ends, the polar head and the non-polar tail. The cell membrane has a fluid mosaic model where it's classified as not rigid or not fixed. Instead, the phospholipid molecules can freely move. So some will sway away and move to another position. Some can also rotate to another position. Some can also move laterally, so within the bilayer. So you can have this phospholipid moving to this end, this also moving to that end, and so on. So they are not arranged in a fixed, rigid manner. No, they are flexible and they are in a form of fluid. So it gives the fluidity of the cell membrane. What helps the cell membrane to become flexible is also the presence of cholesterol. The yellow structures you see in between the phospholipids, they are cholesterol molecules. And the cholesterol molecule also help give the plasma membrane its fluidity. Aside the lipid component, we also have proteins that make up the cell membrane. We have two types of proteins. We have the peripheral and then the integral protein. Peripheral and integral protein. The peripheral proteins sit on the inner or outer surface of the membrane. So when you pick, for example, the green structures you see here, they sit on top of the cell membrane facing outside. And most are also attached to the head. So since it's sitting on the outside, meaning it's attached to the heads of the cell membrane, the phospholipids. Most are glycoproteins. Glycoproteins meaning the proteins are attached to a carbohydrate molecule, and therefore it functions to adhere to some particular molecule and also serves as cell recognition. We also have integral protein where these proteins run across the whole plasma membrane. So it spans the whole plasma membrane. Just like these ones, we have this, what I'm showing you with a pointer. So it has one end extending to the external, one to the internal. So it runs across the cell membrane. Same for this, that, this, that, and that. So they span across the entire cell membrane compared to the peripheral where it's attached to just the upper or the inner part of the cell membrane. We have types of membrane proteins. You can talk of channels. Channels are proteins that transport ions. Channels are proteins that transport ions. And we have carrier proteins that also transport some other molecules that cannot easily pass through the cell membrane, which are not ions. We'll explain more as we move on. Then we also have receptor proteins that upon a ligand or chemical binding to them, they change their shape and then enable that chemical to enter the cell. So basically, they also help with transport. Then we also have enzymes that also speeds up the rate of chemical reaction within the cell. Some also help with transport. We'll talk more about them. Any question up to this class? Where is the classroom? Let me see.
Sister, please, I'm here.
Yeah, Edward. Yeah, any question? The class is too quiet. Or should we conduct a quiz?
Sister, please, no one has raised their hand.
Yeah, so that means it's well understood.
Yes, please.
Hey. Okay, so someone is asking for a better explanation on the carrier and receptor. So a carrier, from the name, it carries substances across the cell membrane. Okay, so the way they are, they just carry substances across easily. We'll talk more about it as we are going to talk of transport. Then receptors, an example of their function is also to transport. When we have chemicals or ligand binding to them, when they bind to them, then it will change the shape of that protein for that particular chemical or ligand to also enter the cell. Okay, we have them. We have different kinds of them. Yes, Williams.
Madam, I want to ask if the glycocalyx is just one structure that we name it glycocalyx, or there are, because the calyx is what is confusing, whether it has subset or something. Thank you.
Okay, so the glycocalyx is not just one. It's just a lot of glycoproteins. Okay, when you say glycoproteins, we have when you say carbohydrate. So the carbohydrate, the cell membrane is also made of carbohydrates. I didn't talk about that. So you have carbohydrate molecules, but these carbohydrate molecules, most of them are bound to either protein or lipid. When they are bound to protein, they are glycoproteins. When they are bound to lipid, they are glycolipids. So we have these glycoproteins that have carbohydrate portions forming a sugary outer coat called the glycocalyx. Okay, so we have a lot of them. There's a lot of these glycoproteins, and that portion that is forming a sugary outer coat, the outer coat is the glycocalyx. So it's not just one. Williams, is it clear? Williams, is he off? All right.
Madam, yeah, it's clear. My mic was off, so.
Okay. Yes, Gideon Kwapna.
Okay, madam, thank you. Please, with the polar and the non-polar, is it the same as the hydrophobic tail and the hydrophilic head?
Okay, so let me say it's not the same. Hydrophobic is related to whether it's water liking or water heating. So whether that part is water soluble or not water soluble. So that is different from the polarity. When you talk of polarity, you talk of the charge. So you have the head being polar, meaning the head is charged, but the tail is non-polar. The tail is not charged. So because of that non-polar nature of the tail, it repels or it prevents charged molecules from entering. I hope that is clear. So polarity is just the ability of that particular molecule to be charged, whether it carries a charge or not. It's different from whether it's the hydrophobic and then hydrophilic nature of the phospholipid layer. Gideon, is it clear?
Yes, madam, please, it's clear.
All right.
Thank you.
Mary.
So it's with the nature.
Pardon?
If I understand, then it has to do with the nature of the phospholipid bilayer.
Yes, it's the nature of the phospholipids that make up the bilayer.
Okay.
That's how it is, yes.
Thank you very much.
Mary Kuma.
Okay, madam. Please, my question is, in fact, it's just a clarification on something. The phospholipid layers. When you you said some cholesterol something, and then you said it gives fluidity or rigidity. I didn't get that. So please, can you?
It makes the cell membrane flexible, more flexible.
Flexible. Oh, okay.
Yeah. So as I said, it's not rigid, it's not fixed. It can somehow flow. Not like water, but the components, the phospholipids, they can, in a way, move around, rotate, move. This one will move to this direction and all that. That flexible nature is as a result of the presence of the cholesterol in there.
Okay, thank you very much.
Last one, Alberta.
Okay, madam, please, I want to get some clarification about so far what you were saying. If I understand correctly, you said the cell membrane has a, like, let's say the structure of it has a form that binds to lipid and others that binds to water. And the main function is that they carry proteins and allow other substance to enter the cells and leave the cell, if I understand it correctly. I don't know if so far that is what you said.
Okay. So you have parts of the where part binds to protein and part binds to lipid is the carbohydrate component. Let me take the structure again. So this is the structure of the cell membrane. A lot of what you see, what mainly make the cell membrane is the lipid form. Okay, it's a lipid. And we also have cholesterol in there. That's also a form of lipid. Then we have proteins. The proteins are what mainly help with transport. I stated some of them. Then we have carbohydrates. So 3 components. We have the lipid, we have the proteins, we have carbohydrates. The carbohydrates, they don't stand alone. It's either they are bound to proteins or they are bound to the lipids. When they are bound to proteins, they are glycoproteins. When they are bound to lipids, they are glycolipids. I hope that is clear. So these 3 components, lipids, proteins, and carbohydrates, make up the cell membrane. Alberta, I don't know if I have answered your question. If it's not clear, kindly come again.
Oh, madam, please, I understand it. Yeah, I now get what you were saying.
Oh, okay.
Okay, thank you.
The time. Last person, let me hear from Georgina. The rest, kindly put it in the chat box. Oh, I think it's only Georgina. Okay. Georgina.
Madam, please, I want to ask if, should in case the actions of the cholesterol is altered here, will the cell die?
The cell won't die, but it's going to lose its flexibility. Is that clear? Georgina. All right, let's continue.
Madam, yes, please, you've answered me. They muted
Sure.
me again.
Okay. So let's look at transport. The transport we'll be talking about is not the transport of substances through the blood to the cells, and no, not that. We're not looking at transport of substances in the blood or supply of blood to parts of the body. That's not what the transport we'll be talking about. This transport is across the cell membrane. So last week I mentioned that almost all functions that we'll be talking about, it starts from the cell, since that's where life begins from. So you want to look at the cell and how the nutrients you get at the end of digestion, the water you take in, the various ions and all that in food, vitamins and all, how do they get into the cell? Because it's the cells that will use them up. When you pick a tissue, it's made of cells. The organs are made of tissue, and when you move down, it's cells. So these cells is what uses the nutrients, the vitamins, the water, ions, and so on. It's the cells that use them during metabolism to help with body activities. So you want to look at how the various molecules are transported across the cell membrane. So the structure of the cell membrane we just saw is selectively permeable. Selectively permeable is when it limits what process. So the nature of the cell membrane is such that it can allow some substances to cross and it will prevent others. So it's a barrier between the outside and then the inside of the cell. It's a barrier. As a barrier, it's like a checkpoint. It allows substances to pass and others it prevents. In between these phospholipid molecules is pores or small holes or spaces. Some molecules, because of their size, very small, they are smaller than that pores, they can easily pass through into the cell or out of the cell. So they can easily move to and fro in the cell because of their small size. Some too, they are larger than the pores in between the phospholipids, and therefore they can't pass through easily. So if they can't pass through easily, then they need a protein, a carrier protein or a channel protein to enable them to cross this barrier. Also, since the nature of molecule please give me layer is phospholipid, as I said, mainly phospholipids. Lipid meaning it will only allow, class, can you hear me? Hello? Hello?
Hello, madam.
Yes, madam. We can hear, but at certain point it
Yeah, you're on
breaks.
it. You're listening.
The line was great.
Please, you want it off.
Eddie, let me control it, okay? When you mute the whole class, you mute me also. Okay. So, or if someone's mic is on, you can mute that person only, okay? Not everyone. For that, you can do it. So, as I said, the lipid layer only enables substances that are also lipid, their nature, to pass through. And those lipid molecules are called fat-soluble molecules, fat or lipid-soluble molecules, meaning they can easily pass through their nature-like structure, which is a phospholipid. So if you are also water soluble, water and fat or lipids, they don't, when you mix, it can't mix homogeneously. The fat or lipid will suspend on top of the water. So here, the fatty nature of this phospholipid repels water, so it doesn't allow water to pass through or any water soluble molecule to pass through. So that is the three factors that influences what crosses the cell membrane. First one, the size. If you are larger than the spaces or pores in between the phospholipid layer, you can't pass through. Secondly, the charge. If you carry a charge, which such as ions, you can't pass through. And also, lastly, your solubility. If you are fat soluble, you can pass through because the phospholipid layer is also fat or lipid. But if you are water soluble or water, you can't pass through. That's why the cell membrane is selectively permeable. It selectively permits smaller sizes, non-charged molecules, and fat-soluble molecules to pass through directly. So substances are able to cross the plasma membrane via a number of different transport mechanisms. And these various mechanisms are classified into two main types: passive and active transport. Passive and active transport. And this difference is as a result of whether that particular movement or transport is needed, or it requires energy. Energy is also dependent on gradient. When we say gradient, we mean whenever there is a difference in a particular concentration of substance in and out of the cell, a gradient is established. I'll take that again. With an example, let's take glucose. If glucose outside the cell, the concentration is higher compared to the concentration inside the cell, a gradient is created. Or when you take outside the cell, for example, you have about 10 molecules of glucose, and inside the cell, you have 5 molecules of glucose. Since there's difference between the two compartments, there's a gradient created. On the other side, if the glucose 10 outside, 10 inside, then that is equilibrium. Equilibrium is reached. But if there is difference in the concentration of glucose, then a gradient is established. So movement of substances from one side of the membrane to the other occurs in the direction of the gradients or against it. We're still using the example of a glucose. Glucose will move from its higher concentration outside the cell into the cell where the concentration is low. There are instances where glucose can also move from its lower concentration to its higher concentration. So if the molecule is moving from a higher concentration to a lower concentration, then there is no energy required because it's moving easily from where it's a lot to where it's low. That is passive transport. Then, if glucose is moving from where it's low to where it's high. Already where it is low. You still want to move it from that low concentration to a higher concentration. You need energy to push it to move, and that is active transport. So within a cell, there are three main types of gradients. You can talk of concentration gradient, electrical gradient, and electrochemical gradient. Concentration is focused on the chemical concentration of solids. Chemical meaning, for example, let's say the number of molecules there. Still using glucose as an example. If you take the extracellular fluid as an example, you have glucose, let's say 10 molecules of glucose. You count them, 1, 2, 3, 4, 5, up to 10. 10 of the glucose molecules. Then the inside, you have 5. You count them, there are 5. That difference in concentration of the glucose gives a concentration gradient. Then we also have electrical gradient. That is with respect to ions. So for example, sodium ions. Outside the cell, you can have, outside the cell, you can have about, let's say 10 sodium ions. Then you have 5 sodium ions inside. Here we are looking at the charges. Sodium is +1. If sodium is +1, then you have 10 plus outside. Then you have 5 plus inside. The difference in the charges across the cell membrane also gives an electrical gradient. The two combined will give you an electrochemical gradient. So the presence of a gradient across the membrane facilitates the movement of substances. As I said, if there's equilibrium, then there's no movement because the inside is the same as the outside. But if there's difference, then that's going to enable movement to occur. So passive, active. Let's take the first one, passive transport. Passive transport is a simple process in which no energy is required to move particles because they are moving from where they are high, a higher concentration, to a lower concentration. Solids or molecules are moving from a higher concentration to a lower concentration across the cell membrane without the use of energy. Across the cell membrane without the use of energy. We have subtypes of passive transport. You can talk of simple diffusion. Simple diffusion is simple, as I explained earlier. If a particular molecule is very small, such that it can pass in between the pores or in between the phospholipid layers, can pass through the pulses in between the phospholipid layers, then that is simple diffusion. Also, they don't carry any charge and they are fat soluble. They can pass through the pores. Simple diffusion. Example is the various gases that we have: oxygen gas, carbon dioxide, nitrogen gas, and so on. All those molecules, gases are small. They have a smaller molecular weight, and therefore they can easily pass through the spaces between the phospholipids. Then we look at facilitated diffusion. Facilitates, help, aid. This type of diffusion uses help, okay? So here the molecule that is being transported, although they are moving from a higher concentration to a lower concentration, because of, number one, their size, maybe they are too larger than the pores, and therefore they can't pass through easily. They need carrier proteins to enable them to pass through. Then also, if they carry a charge, as I said, ions cannot directly pass through the phospholipid layer. They also need proteins called channels. Then you also have water-soluble substances. Water-soluble substances, as I said, since they are not fat or lipids, they can't dissolve within the phospholipid, or they can't pass through easily. So any water-soluble molecule cannot pass through, and therefore they need carrier proteins. Water itself finds it difficult to pass through the phospholipid layers, and therefore they have a special channel called aquaporins that transport water across the phospholipid layer. Aquaporins. So osmosis, also a type of passive transport, because you are transporting only water molecules from a region where water is high, that's in a low concentration, to a region where water is low, that's a higher concentration. Here, we are transporting only water molecules from a region of lower concentration to a region of higher concentration. Here, from a lower concentration, because when you take a lower concentration, you have a lot of water than the solid. That's why we are moving water from where it's a lot to a higher concentration. When you say a higher concentration, we have a lot of the solute compared to the water, and therefore you can't move water from there. The water will move from where it's high, that's from a lower concentration to a higher concentration. Or you can still say moving water from a higher water concentration to a lower water concentration. All same, you are correct. Moving water from a higher water concentration to a lower water concentration. Various mechanisms within the body uses osmosis to transport water across the cell membrane. Example, during formation of urine, absorption of water, and so on. Then we look at active transport. Active, as I've explained earlier, is more demanding because the molecules that you are transporting, first of all, where they are found is low. Their concentration is low, but you still want to move it from where it's low to where it's high. Without energy, you can't move it. Without a push, you can't move it. And that's why ATP comes to play. So here, energy is required to move substances up or against their concentration gradient. So it's also through a semi-permeable membrane or across the cell membrane. Then the energy is in the form of adenosine triphosphate. You have subtypes of active transport. You have primary, secondary, and then vesicular transport. Any question up to this point? I hope the class is active. You are not sleeping. Class, any questions? All right. From the chat room, the class is very lively, and that's good. So I've put them in writings. I've explained them here for you to easily understand all that I'm saying. So also go and look at hydrostatic pressure and osmotic pressure. We'll talk more about it later, but just go and find out the difference between the two. Talk more about it. Osmotic pressure, when you want to talk of osmosis in details. Let's look at the subtypes of active transport. Primary active transport. Primary active transport. Primary meaning that's the basic. Okay, basic. Primary active transport is the basic one because it makes use of ATP direct. It makes use of ATP direct use of ATP. And since most of these active transport requires ion are involved with ions, and so other proteins, other molecules moving against their concentration gradient, they require carrier proteins or channel proteins. So the carrier proteins here, most of them are co-transport. Okay, so co-transporters. Here, when you say co-transport, meaning they transport two. So based on that, you can have some types of transporters. You can have the antipod, you can have the unipod, you can have the sympod. Antipod is just a type of transporter that is transporting two molecules in opposite direction. So when it's transporting one inside, one is also moving outside, antipod in opposite directions. Unipod is transporting just a single molecule. And then we have the sympod. So unipod is not an example of a co-transport. That one is just transporting only one molecule. So it's either antipod or sympod. Sympod is also transporting two molecules, but they are all moving in the same direction. Antipod, the two are moving in different directions. Sympod, the two is moving in the same direction. To keep the class lively, let's break into rooms quickly. You have 4 rooms. First room, when you find yourself in the first room, you are going to explain how sodium potassium ATPase or sodium potassium pump works. How does it work? That kind of protein, how does it transport sodium and potassium? How? Room 2, you also to look at calcium ATPase, how it works. Room 3, proton pump, and room 4, hydrogen potassium ATPase. So I'm breaking you into rooms. Those are sleep, wake up, and then join your individual rooms. Discuss it within 15 minutes, then I'll close the room so that you can join. We all can come together and then discuss them. Take note, room 1, sodium potassium pump. Room 2, calcium ATPase. Room 3, proton pump. And then room 4, hydrogen potassium ATPase. So should your network take you off during the discussion, when you join the class, you can't join the rooms again. So you have to stay in there till they all finish for them to join the main class. So don't worry, if your network takes you off from your individual rooms, when you come in, you can't join that room again. You have to wait till it's done.
Yes, please, we can hear you. And we have someone actually to present on our behalf, and his name is Prince.
All right, let me hear from you, Prince.
All right, thank you, madam. So ours is on sodium potassium pump. So specifically, we are still dealing with fluid compartment, and we have the intracellular fluids and we have the extracellular fluids. So within the intracellular fluids, the most dominant cations are the potassium, and outside, that's the extracellular, let's say the interstitial fluids, the most dominant cations is the sodium. So specifically, as its name implies, sodium potassium pump. So as an antipod mechanism, it's transports those anions in that opposite directions. So one is sending in and one is sending out. So because sodium is really dominant outside, that's the extracellular fluids, the sodium potassium pumps helps to pump sodium inside the cell and the potassium out from the cell. So to maintain the osmotic balance so that it doesn't lead to either shrinkage or swelling of the cell, this sodium potassium cell membrane helps to balance this equilibrium using the ATP mechanism.
Prince, how does it work? Room 1, you didn't answer the question. Anyone from room 1? Friends? Hello?
Yeah. The sodium potassium pump.
Let me mute it.
Friends, I'm listening.
Okay,
Hello?
madam. This pump it binds to 3 anions from outside. Is it my network or what? Hello?
We can hear you.
Hello?
Friends, we can hear you. Hello?
Hello?
Friends, we can hear you. I think there's a challenge with him. Someone else from the group should continue.
Hello.
Yes,
Hello,
who is
ma'am.
this?
Bright Opokwa.
Yes.
Okay.
Yes,
So
Bright,
as
let me.
yes, ma'am. Okay. So as my brother was saying, the sodium potassium pump is meant to maintain the osmotic pressure, and it does that by first, we have the potassium ions being the major cation in the intracellular compartment. And then we have sodium ion being the major ion in the extracellular compartment. But that doesn't mean that we don't have some amount of sodium inside the intracellular, and also potassium. There's some amount of potassium on the other side of the cell membrane, which is in the extracellular compartment. So for balance to be maintained, sodium will have to leave the ICF to the ECF, and then some amount of potassium has to leave the ECF to the ICF. So what happens is that 3 sodium ions will bind to the cell or to the pump. And then when this happens, there is hydrolysis of ATP, and ATP is hydrolyzed into adenosine diphosphate with a free phosphate ion. And this free phosphate ion attaches itself to the pump, thereby changing the shape of the pump. So when the shape of the pump is changed, it allows the sodium ions, the 3 sodium ions that binded to the pump, escape from the membrane to the extracellular compartment. Now, as this happens, 2 potassium ion, also from the extracellular fluid compartment, will attach itself to the pump. And then the phosphate, which bonded with the pump to change the shape of the pump, is released. And as the phosphate is released, the shape of the pump returns to its original shape, allowing the potassium to also escape into the intracellular fluid compartment. And this is done to help maintain the pressure on each side, or the electrochemical gradients on each side of the cell membrane.
Well done. Well answered. Your name again?
Right, Opokwa Pejema.
Right, Opoku. The first guy was Prince Opoku, right? Okay. All right. So that was the explanation I was expecting. I wanted to know in details how the sodium potassium ATPase work, and that's what you have explained. So the pump is also an enzyme. So from the name ATPs, it's also an enzyme that breaks down a particular molecule. Here, ATP. So first of all, you have a lot of sodium is outside the cell compared to the inside. You have a lot of potassium also inside compared to the outside. So if we are moving sodium and potassium against its concentration gradient, meaning we are moving them from their lower concentration to their higher concentration. So you move sodium from the inside where it's low to outside where it's high. You move potassium from the outside where it's low to the inside where it's high. So first of all, the pump is going to carry 3 sodium ions from the inside of the cell. We have binding sites for this molecule. So we have 3 binding sites for sodium and 2 binding sites for potassium. So sodium ion, 3 molecules of sodium ions will bind to the pump. Then ATP will bind to each region, and you will have the enzyme part of this pump breaking down ATP into ADP and then inorganic phosphate. Now, when that breakdown happens, it changes the shape of the pump, opening to the outside, to the external or to the extracellular region. When it happens that way, there is a release of the 3 molecules of sodium into the extracellular space. Then we have the release of the ADP and then the phosphate group, or the detachment of the ADP and phosphate group from the pump. And that's going to bring back the pump to its original shape. But before that, as the pump is facing the external, 2 molecules of potassium will also bind to the pump. And when the ADP and phosphate detaches itself to it, it moves back to its normal shape facing inside. When that happens, potassium is then released into the cell. So that's how this pump carries 3 sodium molecules to the outside and 2 molecules of potassium to the inside. And this is the main carrier protein, or this is the main pump that ensures the resting membrane potential of the cell membrane. Room 1, well done. Room 2. Room 1, kindly lower your hands. So I have 2, Prince Opoku and Bright Opoku. Room 2. Oh, Bright. Is it Bright Opoku Epiyoje? Bright. Bright Opoku. Hello.
Hello. Yes, ma'am.
Is you just answered the question.
Yes, please.
Oh, but you answered the question last week. I wanted someone new. I just went through the list for last week and I saw your name. Okay, no problem. So subsequently, let those who have not yet answered or contributed in class do so that they can also get some scores. All right, room 2. Who is to present? Room 2 or anyone? Calcium ATPs. No one from room 2?
Hello, madam.
Yes.
Yes. So room 2, we are presenting on calcium.
Please, who is
So,
this?
Williams.
Williams, okay.
Yeah, we are presenting on calcium, how calcium is moved out of the cells. And in the first instance, that
How
mechanism,
calcium is moved out of the cell?
like the presentation is on calcium ATPase.
Calcium ATPase. Yes,
Yeah,
the
calcium
activity of
ATPase.
calcium ATPase. Yes.
Yeah. So the calcium ATPase is an enzyme, just as my brother said. The calcium ATPase is an enzyme, and in the cell, when the body senses that there is too much calcium in the cell, the body has to find ways and means to take some of the calcium outside the cell into the extracellular fluid, which is into the blood. So in this instance, we have ATP, which is energy. The ATP also binds with the ATPase region on the calcium pump. And once it binds to it, it causes the pump to adapt to different shape. That is the phosphorylation. So when it happens that way, it adapts a different shape and then helps the calcium to move out. This one, the cell is only moving the calcium out from the intracellular into the extracellular fluid compartment, and then there is a balance. So it will continue to do that till there is a balance between the extracellular fluids calcium and then the intracellular fluid calcium. So after it has been able to move it into the extracellular fluid, it has to come back again, because if it doesn't happen that way, then it means that we are going to move too many calcium outside from the intracellular into the extracellular fluid, and then there will be a deficit again. So in a way of preventing more calcium from going into the extracellular fluid, it has to detach itself. ATP has to detach itself from the region that it binded itself to. And in this way, once it detaches itself, the pump is deactivated and then there is equilibrium. Thank you, madam.
That's what the whole room discussed, and as you all accepted. Williams?
Madam. Hello, madam.
Yes. Did you discuss? Because I was in your room, and your discussion, you were talking of calcium and then ATP in muscle contraction, something like that. And it was way different from what I'm expecting. And from what you just said, meaning calcium is higher in concentration within the cell, which is not true. Okay, from what you just said. And also, movement of these ions is not to establish equilibrium, no. At the resting membrane potential of the cell, when the cell is resting, meaning the cell is not active, physiologically, normally, you don't have equilibrium of the various ions, no. Always, one side of it is of higher concentration compared to the other. And that is normal to the cell. Williams, are you listening?
Yes.
Yes. So first of all, you are not transporting calcium to ensure that the inside and outside is the same. No. Physiologically also, calcium is a lot in the outside than the inside. And movement is from the inside to the outside, all right, because of course we are talking of active transport, and therefore we are moving from a lower concentration inside to the outside, which is higher concentration. Okay, so take note of this. We don't establish equilibrium when we are transporting ions. No, transport is needed.
Okay.
To ensure that the resting membrane potential of the cell, to ensure that always calcium is higher concentration outside than the inside. And that's how the cell is. That's how the various fluids are. That's how it is. Okay.
Oh, okay.
Calcium ATPase. Calcium will bind to its binding site on the pump. Then ATP will come and bind. Then the enzyme part of the pump is going to break down the ATP into ADP and then an inorganic phosphate. When that breakdown happens, it changes the shape of the pump, exposing the outside or facing the outside. Then release the calcium to the outside. Then detachment of ATP and inorganic phosphate would move the pump back to its normal state. That's facing inside. And here we are moving the calcium from the inside where it's low to the outside where it's high. And that's how the calcium ATPase work. I believe you are clear.
Yes.
All right, well done. You still have your score, but you should take note of the calcium ATPase well. Okay. All right, thank you, Williams. Room 3. Room 3. Hydrogen ATPase or proton pump. Hydrogen ATPase or proton pump.
Good evening, madam.
Yes, good evening.
Please, I'm Richard Manfo, and I'm speaking on behalf of Room 3. We are talking on proton pump, how it works. And then basically what we came up with, a proton pump actually helps in the active transport of protein ions across a membrane or a plasma membrane using energy. And then basically what it does is when you take into consideration the stomach, we all know that we have the parietal cells located within the stomach, which is responsible for secretion of hydrogen ions. So it pumps hydrogen ions from the parietal cells into the stomach in exchange of potassium, thereby making the stomach remain more acidic. So basically, that's what proton pump does.
Okay, Richard. Well done, but the question is not answered. Any other person from the group? How does the proton pump work? How? I understand all the explanation you are giving, but you want to look at the cell membrane, across the cell membrane. How is protons transported? Yes, any other person from the group? Edward, are you also part of the group? Edward,
Yes, sister.
Come in.
All right, sister. So with the hydrogen proton pump, this is what happens. The pump uses energy from the ATP, which is adenosine triphosphate, and then it is being broken down into the ADP, which is an inorganic phosphate, releasing the energy. And then it is an active transport in its sense. And then whenever there is a sensation of inadequate hydrogen, as my brother said, the cell uses it hello.
Yes, we are listening.
Hello.
Yes, Edward.
Hey, networking. Hello, sister. Can you hear me?
Yes, we can hear you.
Yeah. So as I was saying, this is an active transport. And then, as my brother said earlier, the cell releases the hydrogen ions, which will make the stomach more acidic for digestion of food.
How?
Hello, sister.
Yes, Edward, how?
Sister, there's going to be a breakdown of the ATP into the ADP.
And what happens?
Actually, sister, I've forgotten what happened, and that there was a term we used within the group. I've forgotten what term we used, but then when the ATP is being broken down to ADP, something happens. That's what I'm trying to recall.
Is it phosphorylation?
Yes, sister. Yes, thank you very much.
Hey, what is it?
I remember we mentioned the term, but then as we were about discussing it, then our time ran up.
Hey, Edward.
Yes, sister.
Okay, all right, that's fine. That's room 3. Edward. Anani.
Yes, please, sister.
All right. So with hydrogen pump, focus on the slide and the pointer. So with active transport, to remind you again, we are transporting these molecules we are mentioning from their lower concentration to their higher concentration. Within the cell, you can see that here the proton molecules are 2, hydrogen 1, 2. Then we have outside the cell 1, 2, 3, 4, 5, 6, 7. So we are having 7 outside, 2 inside, but we still want to move these hydrogen ions from their lower concentration here to the outside where there are many. That's why it's under active transport. And it's a primary active transport because it's directly using ATP. So here, this pump, we have a binding site for hydrogen ion. So hydrogen will first of all bind to it. Then ATP will bind to its binding site on the pump. Then the enzyme nature of the pump is going to break down the ATP into ADP and phosphate group. As breakdown happens, energy is released to change the shape of this pump for the inside here to face outside. When that happens, hydrogen ions is going to be released into the external. Then there's detachment of adenosine diphosphate and phosphate, ADP and phosphate, from the pump. So detachment is going to move the pump back to its original state, facing inside. And that is how this proton pump works. So always hydrogen ion binds, ATP binds, breakdown of ATP is going to enable the pump to change shape facing outside, releasing the hydrogen ion. Then detachment of ADP and inorganic phosphate is going to move the shape back to its normal state. And then the whole process starts again. That is how this proton pump works. So this enables, as you said, more hydrogen ions to be pumped to the external during digestion for gastric digestion to occur. All right. Let's take the last rule, then we end the class here. The last rule, rule 4. Rule 4, yours is on hydrogen potassium ATPase. Hydrogen potassium ATPase. Room 4. Yes, anyone? Do you have a presenter? Room 4, do you have a presenter? Or I can call anyone.
Hello, madam.
Yes.
I'm Frank Safu Jesse, representing room 4, and we are presenting on hydrogen potassium pump. So basically, the hydrogen potassium pump is an enzyme found mostly in the parietal cells of the stomach lining, and what it does is it's going to move hydrogen ions from inside the cell to outside of the cell, which is very abundant the outside the cell than inside the cell. And like you mentioned earlier, that potassium is the most abundant ion inside the cell. So it will move, whereas it's moving hydrogen ion from inside the cell to the outside of the cell, it will also move potassium from outside the cell into where it belongs, which is inside the cell. And it's going to use energy, the ATP. And what's the main reason for this is it wants to create the acidic environment for the stomach to aid in digestion and also to prevent harmful bacteria from thriving inside the stomach. Okay, so inside the parietal cell, carbon dioxide, it combines with water to form carbonic acid, which then breaks down. It breaks down into hydrogen ions and then bicarbonate ions. But what is going to move is it's only the hydrogen ions, because it's moving outside of the cells to create the acidic environment inside the stomach lining. So this is an antipod type of transport because it's moving two molecules, two ions, and in opposite directions. So hydrogen ion is moving from inside the cells to outside of the parietal cells, whereas the potassium is also moving from outside the parietal cells into the cells. And like we all know, moving from a region of lower concentration to a region of higher concentration for the both of them, because hydrogen ion is very abundant inside the stomach, inside the cells, like it's outside the cells of the stomach. So it's moving from inside the cell to where it's very abundant, where it's very plenty. And also potassium moving from outside the cell into the inside where it's also very abundant. So it will require energy because it's working against a concentration gradient. So after the hydrogen ions, carbon dioxide has been combined with water, it will dissociate, it will break down and then form bicarbonate ions and the hydrogen ions. So the energy will move the hydrogen ions only, and it will leave the bicarbonate ions inside the cell. So the energy will move it from inside the cells to outside the cells, and then it will also move one potassium ion from the outside of the cell into the cells to replace the hydrogen ions that has already been moved into the stomach lining, which to cause high acidic content of the stomach. So that is what we found in the hydrogen potassium pump.
Yes, well done, Frank, Jesse,
Yes.
well done. So I think you are all getting it. And why they are all active, all these examples are active transport. Hydrogen, potassium, ATPase, yes. Active transport because hydrogen will move from its lower inside to outside where it's a lot. Potassium will also move from its lower outside to the inside where it's high. So same in the presence of ATP, hydrogen is going to move to the outside and then potassium will move to the inside of the cell. Take note that all these movements or transport we are talking about is not to establish equilibrium, no. It's to ensure the resting state of the cell and some also to ensure other activities like digestion. All right. Any questions up to this point? Any questions up to this point? Then finally, let's talk about secondary active transport, then we end the class. So with secondary active transport, you don't use ATP directly. You don't use ATP directly. Here, the primary active transport will take place and then establish an ion concentration gradient, and that will cause the passive movement of a particular ion down its concentration gradient. When that is happening, it's going to pull along another molecule against its concentration gradient. I know you are confused or you are thinking about it. Analyze it for some seconds. I'll take it again. With secondary active transport, it doesn't make use of ATP directly. However, when primary active transport occurs, you have some ions that will move passively from their higher concentration to their lower concentration. With that same protein moving that particular ion, is going to move along another molecule from its lower concentration to its higher concentration actively. Example is the movement of glucose from the lumen of the intestines into the epithelial cell lining. Here, we have the primary active transport, mainly sodium, potassium, ATPase, transporting 3 molecules of sodium from the inside of the cell to the outside of the cell with the help of ATP. Now, there's a higher concentration of sodium outside, and therefore, using this simple protein, sodium will easily move from its higher concentration here to its lower concentration into the cell. As that movement is happening, it's going to power the movement of glucose from its lower concentration in the lumen to its higher concentration in the epithelial cell. I believe that is clear. That's a common example of a secondary active transport. Sodium moving passively into the cell from its higher concentration to its lower concentration. Then it's going to power the movement of glucose from its lower concentration to its higher concentration into the cell. Another example is as hydrogen ions move from its higher concentration outside to its lower concentration inside, it's going to actively move sucrose from its lower concentration outside to its higher concentration inside the cell. Higher concentration of hydrogen is as a result of this primary active transport, moving hydrogen actively to the extracellular fluid. Analyze this, think about it, read more, watch videos, and better understand. Any questions for the day? Yes, Abdul.
Okay, thank you very much, madam. Yeah, hello.
Yes, you're welcome.
Please, mine is not a question. I just wanted to let you know that I might not be very active answering questions when you ask, but I might be very active at the chat box. So if you are giving extra things, check those of us in the chat box.
So
Yes.
I want to hear your voice too as well. I know
Yes, okay.
the chat box is very active, but once a while, let me hear you, like today. What's your full name,
Yes,
Abdul?
madam. Abdul Waris. I was typing because I was in hurry. I couldn't add the Waris, so I will add it next time.
All right, Waris.
Thank you very much.
You're welcome. Any other contribution, suggestion, question? Alberta.
Okay, madam, please, I wanted to find out when we were talking about the protein pumps inhibitors, you made mention that for the group discussion, when they were presented, they made mention that hydrogen is produced, is moved out of the cells into the parietal cells of the stomach to aid in digestion. And if I understand correctly, in cases of people who are peptic ulcer patients, because of the acidic nature of their stomach, they go into pain and other things when acid is produced in excess. So, madam, I want to find out, in their situation, would the hydrogen still, the enzyme still be released for hydrogen to be produced in order for it to move from the cells into the stomach lining to aid digestion? Meanwhile, it can cause them pain and other things. Please, I want to some clarification on that. Thank you.
Okay. This is a more clinical question. And when you ask a clinical question, you are moving away from physiology, but I'll still answer. So with peptic ulcer is, yes, there is a production of more acid, or not necessarily more acid, but when there is the presence of the gastric acid is there without any work to do, it tends to digest the protein, the lining of the stomach. Okay. Aside that, we have the H. pylori, the bacteria that also causes that. So when in the case where there is secretion of gastric fluid within the stomach, there is production of this H plus under normal healthy state. The cells are secreting the H plus. But the point where it causes the ulcer is the point where there's no protein for this acid to digest. This gastric acid digests proteins. So if there's no protein for the acid to digest, then it's going to rely on the protein of the body made of this, the stomach and then the muscles. So if the gastric doesn't get any, as I said, doesn't get any protein to digest, that's where it rely on the lining of the stomach, eventually causing the ulcer. And normally, as you are there, always there's hydrogen pump working to ensure that the environment within the stomach is acidic, because that's physiologically, that's how it is, so that it digests food normally. So if you are not eating, that's where it tends to work on your proteins of the body. That's the stomach, the lining of the stomach. And that's where it causes the ulcer. So in line with the secretion of hydrogen ions, that is it. But the other cause of peptic ulcer, which is the bacteria H. pylori. Alberta. Alberta, are you good?
Yes, madam. It's okay. Thank you.
All right. Any other, Raymond? Then finally, the leader, Granting. Raymond. All right, let me hear from Philida. Philida, you can't unmute?
Yes, madam. Please, I've been unmuted. Thank you. Please, with the explanation of the
Hello?
yes, please, madam. Please, can you hear me?
Yes, I can hear you.
Yes. Please, you gave an explanation concerning the secondary active transport. Yes. You gave two examples that one molecule is being moved and then it takes another molecule along. So I want to ask, so in short, can you say that as an active, sorry, as a secondary active transport is going on, then there is an active transport also being attached to it. No, sorry. As a passive transport, yes, I think that's the scenario you were giving. So as a passive transport is happening, then an active transport is also happening at the same time. I want to get that clear, please.
Yes, yes, you can say that, but we have these specific proteins that are transporting specific molecules. So it's not always the case where passive transport will also carry a particular molecule moving from low to high. No. We have proteins that are only transporting under passive occasions, moving from high to low. And we also have carrier proteins like these that are also transporting molecules alongside passive, as you have said. So yes, passively molecules are moving from their high to low, and as that is happening, it takes along a particular molecule moving from low to high. That is it. Um, who was that? Philida, are you okay?
Madam, yes, please, I'm okay. So that is to say that for that to happen, I mean, this scenario to happen, it is like a molecule specific.
Yes, all that you are talking about, they have their specific carrier proteins. Glucose would have a specific carrier protein to transport it. Amino acids, they have their specific ones, and all others. So ions would also have their specific molecule carrier proteins that transport them. Okay, so all these that you are talking about, they are carrier protein specific with respect to their various molecules.
Okay, sister. Thank you very much.
You're welcome. Patrick Ofori Techi.
Hello.
Yes, Patrick.
Hello, madam. Good evening.
Evening.
Sorry, please, when you were sharing the rooms, I was in group 4, but unfortunately, I think my network was failing, and then I had to drop off from the group. So I'm asking whether you couldn't join the rooms. What will happen to us, please?
Okay, no problem. So there'll be times where you'll be having these breakout rooms. So you try and then join. You try and then join, okay, to have the
Okay.
discussion another time so that you can also get some scores.
Wow. So meaning today we wouldn't get any scores for today.
No, please.
Oh. Madam, if you beg, it's not fault of ours. It was the network
I
issue.
understand, but it's part of what you opted for. This is an online program. You get it, and
Yeah.
online meaning you should be online during classes. It's not my fault that the network is that way. It's not your fault also, but you try. Okay, another time. Definitely, there'll be times where the network will do you good.
All right. Thank you so much.
All right, you're welcome. Last person, my time is almost up. Raymond, your hand is still up.
Good evening, madam.
Yeah, good evening.
I wanted to throw more light on my sister who asked a clinical question concerning the peptic ulcer.
Okay.
Rather that if someone is diagnosed with a peptic ulcer, most of the treatment regimen, they will give you an antacid, give you an antibiotic, give you something like a proton pump inhibitor like omeprazole.
Yeah.
Then those kind of stuff. So the omeprazole will inhibit the proton pump that we just discussed. So the hydrogens will be moving as we have discussed.
Yeah.
And if the ulcer was caused by the H. pylori, the antibiotic too will deal with it.
Yeah.
So that's what I wanted to add.
Yeah.
Thank you.
That's true. Well done. Well done. Okay. All right. Emmanuel Ochre. It's almost time. So I think
Yeah,
we
madam.
can't address. Yes, Emmanuel.
Yes, madam.
Yes, I'm listening to you.
Please, I wanted to ask a question concerning I think my sister wanted a clarification on it, but your explanation didn't really tackle how, in case a person is given this PPI, like omeprazole, how is it going to affect protein digestion? Yes, okay.
I think Raymond just explained further. Going to inhibit proton pump. That's it all.
Yeah, because mostly, I think we have advised that we should wait 30 minutes before, after giving this medication before we take in food. So I really wanted to see the link why we should wait 30 minutes before, and then the effect of this medication not really creating the acidic environment for the enzyme pepsin and so to act on protein in the stomach.
Okay, so you are still asking a follow-up question on that?
Yes, please.
Okay, all right. So the PPI, as we all know, inhibits these proton pumps. So it reduces the acidic content within the stomach. So when you take it, some also I know some also who also recommend that you take it 30 minutes after eating. 30 minutes you take it. So all these, we are trying to reduce the acidic content or the environment within the stomach. And food intake, yes, is going to go through a series of process and a series of digestion before chemical digestion and so on before it gets to the stomach. So when it gets to the stomach, already we have quite a sizable volume of acid within there. And when the food gets in there through the nervous system or the nervous system pathway, is going to then trigger the production of a required volume of acid to digest the food. So 30 minutes before you take in food, yes, the gastric juice is going to be reduced. But in as much as the food gets into the stomach, physiologically, you have the nervous system to come to play to send signal to the parietal cells for now production of hydrochloric acid to digest the food specifically. So here, you don't have a lot of the gastric juice in the stomach to cause you harm. But when the food gets in there, it's going to trigger a reflex that would enable production of acid to digest the food that has come in. I don't know if I've answered your question, Emmanuel Oche. Hello. So my time is up. It's past time. So we'll end here. Read on the assignments given. If you have any further questions, just put them down and we would address them, God willing, next week. All right. Enjoy the rest of your evening.